Well, good afternoon, everyone. Welcome back to the keynote session and last session of the 2021 symposium. My name is David Rueda. I'm a professor at Imperial College and member of the London Institute of Medical Sciences. And it's my distinct pleasure to introduce the 2021 keynote speaker, Professor Patrick Cramer from the Max Planck Institute for Biophysical Chemistry in Munich. Patrick has always been interested in transcription and control of gene expression from a structural point of view. Already during his PhD in Grenoble with Christopher Müller, he worked on the structure of NF-kappa B and GAMBIF1 complexes. And then he moved to Stanford to work with Roger Kornberg, where he solved the structure of RNA polymerase II, which earned Roger Kornberg the Nobel Prize in 2006. In 2001, he started his laboratory at the LMU and got tenure in just 3 years, and became director of biochemistry in 2010. In 2014, he joined the Max Planck Institute and became director of molecular biology there. His independent group has been extremely productive. It's a pleasure always to read his papers, very insightful. He continues to elucidate the mechanism of transcription and by solving structures of the RNA pol-2 complexes, pre-initiation, elongation, mitochondrial, and in complex with other cofactors such as splicing cofactors and translation. And most recently, he's also been working on protein structures from SARS-CoV-2. Patrick has received numerous awards. I couldn't cite them all, but just to note a few, he's a member of the German Academy of Sciences, he's a member of EMBO, Academia Europaea, and he has also received the Koch Medal of Honor, the German Cross of Merit, the Arthur Burkhardt Prize, and the Centenary Award from the British Biochemical Society here in the UK. Patrick, it's really an honor to welcome you and introduce you. We're looking forward to your talk, and without taking any more of your time, please go ahead and share your slides.
Well, thank you so much, David, for the kind introduction and also for the invitation to give this keynote lecture. I was able to listen to about half of the talks today, and I really enjoyed it. Great variety of technologies, and it's incredible what becomes possible. And hopefully we will also soon, thanks to science and the vaccination, meet again in person. I'm always very happy to go back to England, where I've been as a student in Bristol already in 1992, and then later at Cambridge for a year in the middle of the 90s. So the idea for today is the following, that I talk about what I promised, but before I do so, I tell you just 5 minutes what happened when the pandemic hit. So this was just beforehand. We had a great retreat also with alumni, and we enjoyed ourselves. And then we thought, what do we do now? We were all at, in home office, and a group of about half a dozen people decided to go back to the lab and to work on the coronavirus polymerase. And the reason is twofold. First of all, we are experts in polymerase structure and function, so that we thought probably that's the best way to try and help with the pandemic. And second, the polymerase is an important target for antiviral drugs. So, we managed in record time by 6 people actually just stopping their projects and working for 1.5 months on this problem to solve the structure of the coronavirus polymerase that you see here, and to get a first model of how it functions. And it's actually quite different from other viral polymerases. It contains this sliding poles element, which is important for the processivity of the enzyme, this green structure here. And you need that because the coronavirus RNA genome is the longest known RNA genome. It's about 30,000 nucleotides. And these small polymerases tend to fall off. And so it has evolved a special element to prevent this. Now we were set to look at antivirals, and what is striking here is that all the common antivirals against viral polymerases wouldn't work for the corona enzyme, and the reason is that it has a proofreading function. And that is also because it's such a large genome, you cannot make errors all the time, otherwise the virus would not be functional. And so the traditional antivirals that work very well for other RNA viruses wouldn't work. And then, as you know, remdesivir came, and it's has been, the first drug approved against the treatment of COVID-19 patients, but it didn't work well. There's several reasons. So for that, I cannot go into all the details, but now molnupiravir is actually another antiviral that is currently in phase 3 trials by Merck in the US. And for both of those, we have provided the molecular mechanism so that you actually know how these drugs work. They are both incorporated into the newly synthesized RNA chain that you see here in red. And in the case of remdesivir, the polymerase doesn't stop, so it's not a strong inhibitor of the polymerase. Actually, the polymerase can incorporate 3 additional nucleotides into the growing RNA, and only then there's a barrier to elongation, because these atoms here at the ribose ring of remdesivir will bump into a serine side chain, but that is not enough to stop the enzyme. If you have high concentrations of NTPs, it will go on, and basically it's not a strong inhibitor, and that's one of the reasons why remdesivir is not such a good antiviral. And then for molnupiravir, this uses a totally different mechanism. So it's incorporated into RNA, and then that RNA is used as a template in a second step. And when this happens, the molnupiravir that you see here in orange that is incorporated now can direct the addition of either an A nucleotide or a G, and in other words, it leads to errors, to a lot of errors. It can base pair both with A and G using different tautomeric isoforms of this NHC base in molnupiravir. And that leads to what people have called an error catastrophe. So the enzyme is not stopped by the incorporation of this nucleotide analogon, but it's rather making a lot of errors, and then the virus is not functional anymore. But both of these drugs have big problems that I cannot go into detail. It also has to do with safety and side effects. So we have started to look for entirely new substances, and we already found some substances by large-scale screening with Herbert Waldman at Dortmund that we are now testing for their antiviral potential. Now, but today I promised to talk about our work towards a mechanistic understanding of the regulation of the genome. And why is that an important question? Well, genome regulation occurs to a very large extent at the level of transcription by RNA polymerase II. And that is the central enzyme, as you know, that makes all the messenger RNA, which then, of course, encodes for proteins. And that regulation really underlies the development of organisms, as well as cell differentiation events and also responses of cells to changes in the environment, including actually the immune response. So when you get vaccinated, there's a lot of changes in POL2 regulation, triggering of a very complex immune response that leads to differentiation events, the generation of memory cells, and all kinds of things. So it's very important to understand how this enzyme is regulated, because it's really at the heart of genome regulation. So as a first step, we tried to visualize the process of transcription by POL2. And this, as was said in the introduction, is work that is based on my postdoc work with Roger. So we started from the POL2 structure, and then in the first 10 years of having my own lab, our group was concentrating on crystallizing many different structures of polymerase with different factors, with different nucleic acids and so forth. And that led actually to the first movie of POL2 transcription. You see here just a little scene of that movie. It's actually a 7-minute movie that is on YouTube and used by many for teaching. And that was very satisfactory because now we really understand how the machine itself is working. But really what the next challenge was, and we were busy with this for many years already, and this is continuing, the next big challenge is, of course, regulation. And so how can you even dare to think about solving this problem? Just to remind you, about 1,800 genes in the human genome encode for transcription factors that regulate RNA polymerase II. You know them. These are all the factors that bind to specific sequence elements in the genome and can then trigger a transcription response at many different genes in the genome. And it's really a very complex process. So the way we approached it was by thinking, instead of looking at these individual factors that may only be important in one cell type or for a certain stress response or whatever, let's look at the central machinery that underlies regulation, because if we can understand those transcription complexes and their transitions that are the target for these hundreds and hundreds of factors, then we have a very solid basis for really understanding how genes are regulated on the transcription level. And so that's what we have been doing over the last years, and that's what's ongoing. That will be the main topic of my talk. And we will use that scheme to get a little bit of structure into this really complex process. And all you have to remember for now is that, when you want to regulate transcription, a lot of that regulation, of course, happens at the beginning of genes. That makes sense because some genes take hours to be transcribed because they have such long introns. So you don't, in order to efficiently regulate, this will happen in the beginning of the gene. There will be some decisive decisions taken. And then once you have taken those decisions, then transcription will continue in a processive manner until the end of the gene in most cases. So here is a review from John Liss about 12 years ago. John summarized the key steps at the beginning of transcription that are important as regulatory targets. So in the beginning, you need to open chromatin around the promoter. That is a major step of regulation because the polymerase machinery cannot assess the promoter otherwise. Then you form what is called a pre-initiation complex, or PIC, and that contains a lot of additional factors, not just the polymerase. And then this machinery is able to open the DNA duplex and to initiate the RNA chain. And then the polymerase escapes from the promoter, but it actually doesn't get far. It will soon pause again and stop transcription in a promoter proximal region. And then it looks for other signals that will exchange factors on the polymerase, release the polymerase into the gene and activate it. And it will then also receive factors that allow the polymerase to go through chromatin, so to pass nucleosomes. Now let's go a little bit through and I give you some examples. We start with opening of the chromatin. So what we did is we solved the first structure of a so-called pioneer factor, which you see here in red color, pink-like color, on the nucleosome. And what we found is that this factor can simply use binding energy to begin to unravel the DNA from the nucleosome. So that was an important concept, that there's factors that can invade the nucleosome and begin to release some of the DNA, so it will be accessible for other factors to bind and stabilize that situation. And these factors have long intrinsically disordered regions that can recruit large machineries. In this case, this is a factor called SOX2, which is one of the famous Yamanaka factors that you need to keep pluripotency in cells. And that will then recruit other factors, and this is the case for many other pioneer factors and transcription factors. And some of the first complexes that are recruited are large complexes that can shift nucleosomes around to free the promoter, that can also evict nucleosomes, and that can modify nucleosomes, acetylate them, methylate them, in order to make chromatin more permissive to transcription. And we looked at some of these large complexes. For example, here the first switch-SNF type of remodeler, which is called RISC in the yeast system. And these enzymes use the ATP in order to translocate along DNA. And as they do so, they can, for example, shift nucleosomes, as is observed with this remodeler. And the way this one works is that, the nucleosomes that flank the promoter region will be shifted in either direction. So one of the nucleosomes, the so-called +1, will be shifted downstream, and the other one will be shifted upstream, because the remodeler can bind in opposite directions to those. And that actually frees the promoter sequence. So now the polymerase can assemble with its general transcription factors on the promoter, because there's a region of free DNA. And this assembly of the pre-initiation complex we have recently visualized in the human system at atomic resolution, or let's say building actually atomic models. The densities are around 3 angstrom now. And that is a movie that summarizes how we think this assembly takes place. So there's a set of small general factors, including the TADA box binding protein that can recruit the polymerase. And then TF2E and TF2F form a clamp around the promoter so that it's prevented from dissociation. And now a large factor called TF2H is binding. This is important for the DNA opening. We will move to that in the next slide, or the next 2 slides. But also you need to know that there's also co-activators associating, in particular, this complex that is called the mediator. The mediator stabilizes this assembly, but it also can activate a kinase that is called CDK7 and seen here in orange. And through that activation of that kinase, the whole complex will be destabilized again, because now after the DNA has been opened, the polymerase needs to start synthesizing RNA, and it needs to escape from this large pre-initiation complex. So this function is also built in with some delay. There will be phosphorylation, and then the polymerase can escape. But what I want to show you is that we not only have the human structure, we also have the yeast structure, and that is actually the one we were first solving, and then later we went on to the human. And it's incredibly striking how well conserved these complexes are. We were expecting that to some extent, of course, based on sequence, but structure is much higher conserved, much better conserved, as is seen in many systems. And it's really quite only for the experts to see differences and to explain to them. There are few, but actually really only few differences. And that tells you about the importance of that machinery, that it has been constant over evolution, essentially constant from yeast to human in all eukaryotic cells. Now, it was good that we had both the yeast and the human systems, because sometimes we could solve a problem in the yeast system and sometimes in the human system. So talking only about the mechanism of opening of the DNA, we could actually trap an intermediate in the yeast system. You will see that when you look carefully, there's about 5 base pairs here in orange that get separated by a few. The bases get separated by a few angstrom with the first power stroke. And then there's 2 loops here from both sides that stabilize that intermediate. And so we could actually trap this early intermediate of DNA opening, which we could not in the human system. But then on the other hand, we could not trap in the yeast system this machine, this ATPase that causes the opening. It's a translocase that introduces strain to the DNA. But in the human system, we could actually see that this ATPase undergoes a 3-step transition. So it binds the downstream DNA, and then it undergoes a conformational change when ATP is bound. And then upon hydrolysis, the enzyme is reset, so that there is a twist introduced into the DNA that is then propagated downstream, and that leads to the initial opening of the DNA. Now, is that at all relevant for regulation? Well, for this you need functional genomics, and we were using labeling, metabolic labeling of RNA in cells. In this case, we used yeast cells, but it works really well also in human. And we were, removing very rapidly from the yeast nucleus this translocase activity that opens the DNA. And we were asking, do all genes depend on this enzyme, or is there differences? And most genes actually need it. So about 80% of the yeast genes will be dramatically down-regulated when we remove this enzyme, this ATPase that opens the DNA. But about 20% of the yeast genes don't really care so much. Transcription still works, there's still RNA being produced. And when you actually check their promoter sequences, they are predicted to be melted more easily. So you see, this is how evolution works. You have sequences in the promoter, which are the initially melted region where the DNA opening starts, and you can evolve that region to be more or less difficult to melt. And that, gives you more or less of a dependence on this factor, on this ATPase. Now, of course, the polymerase needs to be recruited to active genes. And the way you have to look at that is that most polymerases are engaged with the genome at any time. And if you want to change the transcription program, the gene expression program, you have to redistribute these polymerases. So you need to bring them rapidly to genes that you wish to activate. And we think that the C-terminal domain of the polymerase, which is a very long intrinsically disordered extension, is involved in this recruitment process. Why do we think so? I show you some slides, some indication for that. But you have to imagine that this domain is about 10 times the length, or its length is about 10 times the diameter of the polymerase. It's drawn here approximately to scale. So, and it's also an extremely highly conserved sequence. So if you just make that CTD, you can form, in recombinant form, you can form droplets in solution. So there's, self-interaction between these CTD molecules. And if you then ask, is this self-association important in vivo? We teamed up with Xavier Darjac at Berkeley, and they could show that when they truncate the CTD in human cells to half of its length, then you get less polymerase clusters, as seen here by high-resolution light microscopy, and you also get clusters of lower intensity. So that really points to a role of that CTD for clustering of polymerases, recruitment of polymerases in vivo. Now, does that mean anything in terms of function? The answer is yes. So what we've done is we've now used those human cells where the CTD is truncated, and we asked whether, when we induce a stress response and we look at the genes that get activated, how does that stress response change? And we found that, similar genes are activated, but the whole stress response is delayed in those cells where the CTD is truncated. And that is dependent on enhancers, on, distal regulatory regions where these transcription factors bind, these activating factors bind, as we've seen with our protocol called TT-seq for transient transcriptome sequencing. This is also relying on metabolic RNA labeling. So I think this is one good example where we have, the function of such an intrinsically disordered region in the recruitment of polymerase. And that is not just, for architectural reasons, it actually has a functional consequence for the cell. So you need this rapid recruitment in order to allow for the rapid activation of genes, because you need to bring polymerases specifically to their target genes and very rapidly. So we're now already, with these few examples that I showed you, we went half the way through this scheme. So we looked at chromatin opening to some extent, and the formation of these pre-initiation complexes, and then DNA opening. And now in the second half, I will come to another very important regulatory process, which is early transcription elongation, something that happens in the promoter proximal region. So as I said, once you open the DNA and the polymerase starts to make RNA, it escapes from the promoter, but it does not get far. It pauses in the promoter proximal region, and then it waits for signals to be activated, and only then can it pass through nucleosomes. So what we did is that we looked at this process of pausing and regulation at the pausing stage in great detail over the last few years. And since we know from functional genomics the arrangement of these complexes in the beginning of genes, we can actually now make a model for this. So the pre-initiation complex is separated from the paused polymerase by about 50 or so base pairs. And then there's a +1 nucleosome just downstream of this paused complex. And since we have structures of the PIC, of the paused complex, and of the polymerase with the nucleosome, we can make a model of the entire situation here at the beginning of genes. And what is striking is that you can fit everything, you know, when you use the distances on the DNA that are known from in vivo work. But you also can predict that this paused polymerase would interfere with new initiation. You can assemble a pre-initiation complex, that's the prediction, but you cannot open the DNA, because if you wish to open the DNA, you need to bring this downstream DNA into the active center of the polymerase. You need to draw it from this position where the paused polymerase sits, and that is difficult. Now, to test this model, we developed multi-omics techniques where we combine different functional genomics protocols, and we combine them also with a kinetic modeling of the whole process. And this is shown here and summarized in this movie. So what we find, so each dot here is, you know, one human gene, transcription from one human gene. And we plot here two kinetic parameters. One is the frequency of initiation, so how often the polymerase initiates. And the other one is the duration of pausing. So how long does the polymerase pause here at this point? And what you see is that when you artificially extend the pausing using an inhibitor of CDK9, of an enzyme that actually releases the pause, then you see that pause duration goes up in cells genome-wide. That is expected. But you also see that initiation frequency goes down. So that, you know, proves our model that when you have a paused polymerase here, that downregulates or impairs initiation. And that was something predicted by Jesper Svedstrup almost 10 years ago, based only on kinetic considerations without data. And it's beautifully seen here genome-wide in human cells. Now, what has come up over the last few years only is that this is only part of the story. So actually what can also happen is that this paused polymerase is destabilized and actually lost from the gene. And this is what we call premature termination or attenuation. A very interesting process. And we have recently investigated this process, and I want to share those data because it's unpublished. And that is really the heroic effort of Isaac, who was able to make the factor that will destabilize and terminate the paused polymerase. And that is a 16 subunit protein that he made by co-expression of those subunits. And he could then assemble the entire 34 subunit complex and look how that integrator complex actually works. So you see here the paused polymerase on the left, and which is the target, right? And then you see this large integrator complex on the right. And what happens when integrator binds the polymerase, there's one subunit, the first subunit integrator, subunit 1, which gets ordered because it now binds to its target in some kind of induced fit. And what is also quite striking is that the integrator will bind not only to the polymerase, but also to two factors that are integral parts of the paused complex. And this is how the integrator actually recognizes the paused complex. It binds to these two factors called DSIF and NELF. And these factors are bound to polymerase when it's stably paused, so it can recognize that state of the polymerase, because otherwise it would target the polymerase everywhere. That would be a problem. You would cause termination at all kinds of places in the genome, which is not the case. So this is the so-called pre-termination complex, where Integrator has bound the paused enzyme, and quite strikingly, it also binds this C-terminal region that I was discussing that is highly disordered. And what happens is that a very, very short part of that C-terminal domain, only 12 or so residues are actually bound by the polymerase. But that is enough to generate some specificity, because that C-terminal region is highly phosphorylated, and it's known from work of others that phosphorylations at these tyrosine residues here will increase the affinity. And it's quite interesting that in our density, these residues point to conserved basic residues in Integrator. So you can easily imagine why these phosphorylations increase the affinity in the first place. But now, what does Integrator actually do? There's one subunit that will actually cleave the nascent RNA, and that is seen in this movie here, where we remove all the subunits except for the one endonuclease, and it really opens up when Integrator binds to its target, and then the nascent RNA can extend into the active center cleft of that endonuclease, where it can be subject to cleavage. And this happens about 20 nucleotides from the active site of Pol II, where this cleavage occurs. And that agrees very well with data from Karen Edelman, who has mapped these cleavage sites in vivo. So we are sure now about this mechanism, because we observe that distance in the structure, and we observe it in vivo also by sequencing. So what is happening then? Well, you can imagine, the RNA, the nascent RNA that extends from the polymerase, needs to be protected. Otherwise, it would be degraded immediately. And that protection occurs, of course, through the 5' cap structure that you all know. And that cap structure is now removed by Integrator, because that endonuclease will cleave off a piece of RNA that contains the cap. And we think that this is basically providing an entry point for exonucleases that can then terminate the polymerase. That is, I think, one possibility, but it needs to be studied. This is all still a rather new mechanism. Anyway, so our scheme looks like this now. You have the pre-initiation complex, the paused complex, and that paused complex will downregulate initiation, but also the paused polymerase, at least a fraction of it, can get terminated through Integrator binding. But of course, when there's a positive signal, so that the gene really should be switched on, then what happens is that you convert this paused polymerase into an actively transcribing elongation complex. And this is seen here in the right upper corner. And we also have the structure of that complex. We call it EC star because it's an activated complex that now carries positive elongation factors, in particular the PAF complex and SPT6, but also other factors. Now, we can do all this biochemistry, and we could find out why the polymerase pausing is stabilized. We could also see why the so-called PTEF-B complex can, or how the PTEF-B complex can convert the paused polymerase to the actively transcribing polymerase. This you can all do biochemically, and you can figure out the mechanism. But in the end, you need to also look in vivo in human cells, what these different factors do, and whether the prediction from your biochemistry are holding in vivo, or what the actual function of these factors in vivo is. And so I give you one example. We've looked at many of those factors, but recently we also looked at SPT6, because there's only really data from yeast, and now we can also rapidly deplete it using chemical biology from human cells, and then using multi-omics, we can monitor what happens to transcription when SPT6 is depleted. And I'm missing that slide. That's a pity. Okay, anyway, what happens then is that the polymerase gets stuck early in the gene. Maybe I have the slide later. Okay, now what is important to realize is, is there a structural mechanism that underlies this switch? So we have here a switch, obviously, where the post complex can either bind integrator and then get terminated, or the post polymerase can be converted into the active elongation complex. And both things cannot happen at the same time. And the reason is quite simple. When you superimpose the two structures, you see 4 major clashes. So integrator cannot bind when the positive factors are there, and the positive factors cannot bind when integrator is there. And the switch between those is really phosphorylation. So this PTEF-B kinase complex will destabilize binding of negative factors, and it will stabilize the binding of positive factors. Now we understand better, how we get to a actively transcribing form of the polymerase. And now we need to, of course, look at the question, how does that complex now go through nucleosomes? Because the factors are being exchanged in front of the first nucleosome. But now you have to understand also how polymerase advances and goes through nucleosomes. And we could recently also elucidate that process to some extent. Actually, now we see the SPT6 data. So we ask, what happens when you deplete SPT6? And we see that then the polymerase has problems to go through the first nucleosome. So this is a technology called MNET-seq, actually first described by Churchman and Weissman, and then adopted by Nick Proudfoot to the human system. And that is a beautiful technology that allows you to map engaged polymerase over the human genome and to see the occupancy of polymerases that actually carry an RNA transcript. And what you see is when we deplete SPT6, then in this region of the +1 nucleosome, you accumulate polymerases. So there's a problem to go through the first nucleosome, which shows you that SPT6 is important for that passage. But there's other factors that are important for the polymerase to go through nucleosomes. And 2 of them we analyzed recently. We did the biochemistry, but we also reconstituted complexes and got the structures. One of them is called CHD1, and the other factor is called FACT. And the way we have looked at them here structurally is quite interesting, I think, in terms of technology. So what we've done is we've assembled a complex of polymerase in front of the nucleosome. And then we have started transcription by adding nucleoside triphosphate substrates. And we let the polymerase run into the nucleosome until, a certain point. You can define that point by having, DNA sequences where, for example, when one nucleotide is lacking, you get the polymerase stalling at a certain position inside of the nucleosome. And the polymerase can enter the nucleosome alone. It just cannot pass through the entire nucleosome. But what happens then is that the polymerase is generating partially unraveled nucleosomes. And those partially unraveled nucleosomes, which have some DNA detached from the histone octamer, these partially unraveled nucleosomes are substrates for binding these factors that help the polymerase to progress through the nucleosome and to make it all the way through. And this is how we could assemble those complexes and how we could solve those structures. And there's many things you can learn about it, but maybe just one aspect. This CHD1 enzyme is again an ATPase, a translocase, and it binds in a way that would pump additional DNA into the active center of the polymerase. So it would help the polymerase to go through the nucleosome. And then this FACT complex is more of a chaperone. It was first characterized by Danny Reinberg, and that can only bind once DNA is partially detached. And the FACT chaperone will actually, that was shown by Caroline Luger recently, it will substitute for some of the lost DNA interactions, and that actually prevents the loss of histones, because polymerase needs to remove those histones when it wants to go through the nucleosome. But you don't want to lose those histones from the site of transcription because they carry information, right? They have certain modifications in the histone tails that are important because they provide a signal that you have here an active gene, and you don't want to lose them. And for that, you need these assembly factors or histone chaperones. Now, finally, the polymerase will go through nucleosomes and it will be able to enter the first intron of the gene. And when it enters the intron, it passes over the 5' splice site. And when it does so, it can recruit the U1 SN RNP or U1 SNRP, which is actually the first particle of the spliceosome that will be recruited by the transcribing polymerase to active genes. And we have recently been able to solve the structure of the polymerase in complex with the U1 SNRP. This is actually the first structure where parts of the transcription machinery have been combined with a part of the spliceosome. And therefore, it's very important for understanding co-transcriptional splicing, and to be more precise, co-transcriptional spliceosome assembly, which is actually quite different from co-transcriptional splicing, which is the actual process of removing the intron. So from these results, we have come up with a model, and that model is based by our observation that when U1 SNRP binds to the polymerase, it not only binds through an RNA contact that you see here, the formation of an RNA duplex on the surface of polymerase, it also binds through a direct protein-protein interaction. And that was unknown and unpredicted. It's quite striking. There's one subunit in U1 SNRP that binds to the surface of Pol2. And that, of course, lets you think very differently about co-transcriptional spliceosome assembly, because you can now suggest a model that we call the growing intron loop model. So what the model basically says is when polymerase goes over the 5' splice site, it will bind U1 as an RNP. But because U1 as an RNP is not just bound to the RNA, but also to the polymerase itself, it could travel with the polymerase further. And that would lead to the formation of this intron loop. This is why we call it growing intron loop model. And what is the advantage of that model? The advantage is that the intron is very, very long, and it has been a mystery how, after the polymerase has been going over, let's say, 10,000 base pairs of DNA, intronic DNA, how it would be able to pair the 5' and the 3' splice site on the RNA. You can imagine if there are 10,000 nucleotides apart in the nascent RNA, that's a logistic problem. But this model solves it quite easily, because U1 as an RNP is recruited, it stays on the surface of the polymerase, it travels with polymerase, and the RNA is forming a growing intron loop. And as the 3' splice site is passed, and the branch site, of course, you can easily facilitate functional pairing of those two splice sites, because the 5' splice site is retained on the polymerase. And now when the 3' splice site appears on the surface of the polymerase, you can assemble what is called a pre-spliceosome, or the so-called A complex. That is our model. Now, we did one more experiment to look at this process by stopping further spliceosome assembly at this stage of A complex formation. You can do that with a drug called PLA-B. And we added that drug to human cells, and we monitored what is happening to splicing and what is happening to transcription and what is happening to polymerase. And something really interesting was found. But first of all, we see that, even after an hour of treatment, transcription is down almost to a half of the extent. You see that by nascent RNAs or newly synthesized RNA signal using TT-seq. And what happens is that the polymerases accumulate in the promoter proximal region. You see that by MNAT-seq. So the issue is that when you prevent further spliceosome assembly, there's some signal that tells the cell something is wrong downstream at the intron. And then there's a signal that tells the polymerase you should stay paused at the beginning of the gene. No more polymerases should begin to transcribe because there's an issue with co-transcriptional spliceosome assembly. Now, how this works over distances is very difficult to understand. It's something we are investigating in the future. But I imagine that these processes are actually very happening, very close together in space, and that both DNA and RNA are looped out from a transcribing enzyme. So this is the summary. This is what I wanted to tell you today. So I summarize briefly. We have tried to serve the community by adding structural information and also functional information on these early steps of Pol2 transcription. So we know now how many of the involved complexes look like. We know about many of the molecular transitions, which factors need to be exchanged, which conformational changes are occurring. And that, I think, provided quite a lot of insights into the mechanisms that are underlying these early steps in transcription, which are the target for regulation. And in particular today, I was concentrating on very recent data on premature termination, which is a new process that could not be reviewed 10 years ago because it was essentially unknown that this occurs to a large extent, and that which the factors are that are involved. And I talked about pre-spliceosome assembly. And what is striking here is that it can happen on the surface of polymerase and that it provides a feedback to transcription. The traditional model is that transcription provides a signal for the splicing machinery, and that is basically occurring downstream. But we see here that the reverse is also true, and that somehow transcription can sense whether splicing is working fine or not, or at least spliceosome assembly. And that is a signal, to that the gene should have full transcription activity. And if something goes wrong, then there's a signal that transcription should be downregulated at the beginning of the gene. So basically, we have evidence for these different checkpoints, and this, is all summarized here on the left. Our current understanding of how regulation works on a Pol2 gene. There's all these activators that influence initiation. They can do that most likely using different mechanisms, recruiting, these modification enzymes, but also recruiting initiation factors and so forth. But then these early and late elongation checkpoints provide a feedback to initiation, and they can downregulate initiation if something goes wrong, or maybe also most likely for regulatory reasons. So we have evidence that I didn't show you in human cells that you can really downregulate initiation this way, or in other words, you can upregulate transcription when you remove the elongation blocks by delivery of this positive elongation factor B. So this is what I wanted to tell you, and just to say that in the future we would like to study more and more of that in the natural context, in the chromatin context, and maybe also going in vivo one day. But I think as an intermediary step, we need to go to larger, higher order structures, for example, parts of chromosomes where we look at active genes. And this needs a lot of development of technologies, of course. So many people have been involved over the years, and I really could only provide an overview, and I cannot mention everyone, but here are really key people that have been involved in what I was telling you today. Sandra, Shintaro, Vasan, Clemens, and Kayo, they have been key people to work on the initiation process. And then Carrie, Seychelle, Lucas, Saskia, and Björn have been key people working on the early elongation, which was done mainly in the human system, whereas a lot of the initiation work was done in yeast and only later in human. Now, Isaac, I told you, is the hero who did the recent unpublished Integrator, a complex structure bound to the post-polymerase. He was helped by Ying, but Ying also did other work on elongation factors. Su Yang did the great work on the U1 SNRP, and she's actually returning to Cambridge. That was just announced. I'm very happy that she got such a great position. She will be, I think, a great colleague for people in Cambridge, and she will follow up alternative splicing, because there's a lot of regulation also on the level of splicing, and alternative splicing is probably to a large extent set or connected to transcription in a way that we don't understand on the mechanistic level. So I think we can expect a lot of interesting insights from Su Yang. Christina, Livia, Sarah, and Michael, they have done functional genomics and bioinformatics on the elongation control. And then Christian is our hero who all the EM projects and is maintaining the facility. And then Henning Urlaub is our most important collaborator, because he's doing all the proteomics, including the identification of crosslinks. And I didn't have time to talk about Haibo's most recent work, but that is something interesting also that should come out early next year. Well, this is what I wanted to tell you, and I thank you again for the invitation. Thanks for listening. I know it was a long day. Hopefully I could entertain you with making you understand some of the principles that underlie the regulation of Pol2 transcription. And with this, if you still have time and energy, I'm happy to try and answer some questions. Thank you so much.
Thank you so much, Patrick. That was fantastic talk and amazing work as always. We're going to give people a few minutes to maybe start typing up the questions in the Q&A. Please don't be shy and ask any question you might have. In the meantime, maybe I'll ask a question or two. I think the structures with the nucleosome and factor are amazing. I didn't quite understand. Is the model that FACT disassembles the nucleosome, or does it stay assembled, or does it just remove the H2H to B dimer? Or what are your thoughts on this?
So it looks from, I mean, we don't have proof for that, but it looks the most plausible model now, taking, you know, the functional data and the structural data together, is that polymerase first enters the nucleosome. It can do that, you know, it can unravel a few turns of DNA, but then it begins to pause. We see that in vitro, but you can also see it in vivo when you remove factors. And then that intermediate that you have then, this, you know, paused polymerase inside of a nucleosome, which is different from the standard paused polymerase, this intermediate can recruit FACT. And the reason is that now histone surfaces that are normally covered by DNA are available because the DNA is detached. And then there's regions, in fact, that can bind to those exposed surfaces and prevent the loss of those histones once the DNA is unraveled. And what then happens, we don't know, but we think that, you know, when polymerase goes further, then the histones are set free, but they are stuck to the site of transcription because they have now this interaction with FACT. So they can be used now to re-establish a nucleosome in the wake of polymerase. And this way that's the model. That's the model. This way you can prevent loss of histones.
Yeah, effectively increasing the local concentration of histone, I guess. Yeah, I
guess so. And especially those variants that are associated with active transcription.
Yeah. We have a first question in the chat by Swati, and she's asking, does FACT bind to polymerase? And do you think they also do conformational changes in polymerase, or does it bind only to histones?
Yeah, that is a great point. We don't observe direct contacts between FACT and the polymerase in our system, but we are still lacking in those structures some of the elongation factors that are always bound to polymerase when it's in vivo. So for example, SPT6. And it's known that SPT6 again binds a factor called IWS1. And that factor has been described to interact with FACT. And so through this, assembly line, we think that FACT is also anchored to polymerase. And this explains how you can retain histones at the site of transcription. But those interactions we don't see in our structures yet because we don't have all the factors in there.
Yeah, absolutely. As people ask more questions, I will ask one more question about the U1 SNRP interaction. I think one of the predictions would be that maybe U1 SNRP should co-purify with RNA polymerase through this direct protein-protein interaction, but I don't think this is typically the case. And I was wondering if maybe this interaction is regulated by other cofactors.
That's a good point. So what you know, I guess, and is that have been several papers which have implied U1 SNRP as an anti-termination factor. So maybe 3-4 papers, and this process has also been called telescripting in the literature. And basically the idea is once U1 SNRP is bound to an actively transcribing enzyme, it prevents the poly-A machinery from coming in prematurely, before you reach the end of the gene. And there's data from Phil Sharp also that this is important for promoter directionality, and from Torben Jensen. So, but whether this is due to direct interactions, I think is not so clear. The co-purification, it's probably difficult because these interactions are transient and because they're probably resolved in actively transcribing cells rather quickly once you form pre-spliceosomes, and then you go on into a B complex where the tri-SNRP is recruited. But I'm not sure, maybe there is some evidence of co-purification that simply I'm not aware of.
I definitely like the model very much.
Okay, we've got another question here, which is, so Mitten then asks, how conserved are the machineries of transcription between bacteria and archaea?
Yeah, very good point. I didn't have time to talk about other polymerases. There's of course Pol1, Pol3 and eukaryotes, and then the archaeal and the bacterial enzymes. The enzyme itself is highly conserved. So it has a core of 5 subunits, which is the same essentially except for a few domains. And then the eukaryotic and the archaeal enzyme have additional subunits, 12, 14, 17 for Pol1, 2, and 3. So the core is the same. But then when you go to the initiation factors, it's quite different. So you have sigma factors in bacteria, but you have these channel factors in the Pol2 system that I showed. And you have different ones for Pol1 and Pol3. And that explains actually how you bind different types of promoters, right? Because that is mediated by these different sets of initiation factors.
Okay, we've got a question from Phil, and he makes a comment saying, mind-blowing talk. And he says, in the light of this amazing complex process with many checkpoints, can you comment on the idea that there's widespread transcription across the genome? In his brackets, he says dark matter RNA, or is this just simply noise in the system?
I think both. It's a very good question. For 10 years or so, we know when you sequence very deeply, you can get RNA from many regions of the genome. So there is some background for sure that the polymerase is not 100% specific to only start at promoters and to only start at active promoters. So there's some sloppiness, but of course, a lot of it is also regulation. So for example, we have thousands of enhancer regions, which are very distant from the gene and then loop in and provide a signal. And those enhancers are known to produce what is called enhancer RNAs. And the enhancer RNAs are very short-lived. They live on average for a minute or two. And so it disappears rapidly. But when you use RNA labeling, you can observe it. And it's actually a proxy for enhancer activity. The more of those RNAs you observe, the more active the enhancer is. So, and then there's, more stable non-coding RNAs, link RNAs, and so forth, which also have sometimes a function in trans. It's known, for example, from dosage compensation, the exist RNA and others. So, yeah. But I guess there's also a lot of background, because the polymerase is a good enzyme. You have to actually prevent it from doing its job. Otherwise it would transcribe, in many places. So when, for example, when you deplete some of the factors that maintain chromatin, then suddenly the polymerase will start in the middle of the gene. So chromatin is actually protecting the genome from a spurious transcription by polymerases that are just keen to transcribe.
All right. Well, I don't see any more questions in the chat. Patrick, it's been fantastic. The one thing I missed from these virtual meetings is a round of applause. So you have a virtual round of applause from all of us.